In this video, we shall continue with the next topic, that is microsporogenesis. Microsporogenesis, it means that it means that it is defined as the formation of the microspores, formation of a microspores inside the anther. So as we have already seen, as we have already learned in the last video, in the last part of the video, we have already seen the structure of a stamen, we have already seen the structure of the anther, and we have already know the labeling, the 4 layers of the anther wall and the microsporangium. So here I have labeled that microsporogenesis is the formation of a microspores, and here I have labeled the 4 layers of the anther wall and the microsporangium. Here, as the anther they are still young, the cells inside the microsporangium are known as the archegonial cell or the sporogenous cell. Sporogenous cells. These are the sporogenous cells. So as the anther starts to become mature, the microsporangia, when the anther becomes mature, the microsporangia, it becomes wider. It becomes wider in size. But the sporogenous cells inside, they stay in the same size, in the same number, in the same mass. It does not divide, or it doesn't multiply yet, but the size of the microsporangia, it becomes wider. So when the anther starts to become mature, the microsporangia becomes wider, and these sporogenous cells which present inside the microsporangia, they are arranged in a loosely manner, that is a space between them. See, because when the anther has not yet matured, the sporogenous cells inside the microsporangia, they are arranged in a tightly manner, but when the anthers become mature, the sporogenous cells is arranged in a loosely manner where they have no contact of each other, and when this happen, this is known as a pollen sacs. When the anthers become mature and the sporogenous cells becomes loosely contact with each other, it is known as a pollen sacs. So it is known as a pollen sac, and these sporogenous cells, here it is known as the microspores mother cell. These microspores mother cells, they are diploid. So these microsporogenous cells, they are diploid. So they are undergoing the process to become a microspores. So the first steps that they are undergoing is, when these microspores mother cells, they are diploid, they will undergo, suppose this is a microspores mother cell, they will undergo the meiotic, they will undergo meiotic division, and meiotic division, it will result in a reductional. So it become reductional. So it will divide and give rise into 4 cells. When they undergo meiotic division, it will give rise to 4. So we will draw all the cells, but which we mostly found. So the first one is the tetrahedral. This is a tetrahedral. And the second one is the isobilateral. So bilateral. And the third one, and the next one is the decussate. One, two, three, four. This is a decussate, or it is also known as the F shape. These microspores mother cells, they undergo their meiotic division and give rise to 4 cells. Now, in the tetrahedral, this is the first, second, third, and 1 at the back. And in the isobilateral is 1, 2, 3, 4, as we can see, and in the decussate 1, 2, 3, 4. But in angiospermic, this is most common. The tetrahedral is the most common. So after some time, so the next steps after they undergo their meiotic division, the next steps is in a pollen sac, the most common is the tetrahedral. So after this, some of them they degenerate, and some they got separated into 1, 2, 3, 4. These are the microspores. And from here they will develop the layers which surround them, and they will divide itself to form a mature pollen grain, which we will study in our next video.